for the search for life. I will try to organize my talk so that we can follow this outline from the vastness of the mere search for life that all of us are interested in, towards the concreteness of using some particular molecular tool so we can have a whole idea about the whole subject. And I will start by talking about the need that we have as human beings to understand life from its origins to its features and peculiarities towards the distribution they have in the world. So since the dawn of mankind, we man and woman have wondered about life from not just an existential point of view, but also from a scientific point of view. And we have wondered ourselves a lot of questions regarding life, such as where did life come from? Where there will be life beyond our planet? What will it be like? And how, will it be similar to the life that we know on Earth? And the thing is that today it is still not clear where and how life arose. But there are instead many hypotheses and theories about whether life came or emerged on our planet directly, or whether it was arising somewhere else in the universe, and that it may have come into our planet. And in the former case, it is not clear either whether it could emerge in submarine events or rather in subaqueous terrestrial hot springs, or maybe rather in volcanic areas when the presence of chemical species essential for life could have existed, or rather in impact zones where meteorites after impact could have created the conditions conducive to life, or even from the energy imputation provided by lightning strike. And connected with this, in the search for life, we also wonder about the very limits of life, because it is widespread everywhere in our planet, and it actually exists in so much varied environments, such as some of them very hostile and in principle not very conducive to life, such as cold and dry deserts, very hot and arid environments that very often contain elevated contents or concentration of salts or toxic elements, aqueous environments with hyperarid or hyperalkaline waters, often contaminated with mine drainages or nuclear residues, for example, subsurface environments subjected to high pressures or high temperature fluids, or even polyextreme environments, which are those that we know that combine several of these severe conditions related with parameters like temperature, pH, radiation, salts, toxic elements, and so on. The way that we address the search for life may also be very varied if we take into account the variety of approaches that the different disciplines involved in the search for life involve. For example, it won't be certainly the same, the approach that a biochemist will follow compared with a paleontologist, for example, a geologist or an astrophysicist, neither in scale nor in the target that they will follow. That the target that a biochemist will follow would be, for example, to search for a molecule, whereas a paleontologist will go for a microstructure, whereas a geologist, a macro texture, for example, or an electromagnetic signal in the case of an astrophysicist. But the thing is that the different disciplines have a common basis, which is the detection, the search for a measurable feature, whether physical or chemical, that may attest the presence of life, whether extant or extinct. We don't care, but we want to know about life somewhere else. And another perspective that may change depending on how and where do we search for life is how do we approach that search. It can be done remotely or in situ. In remote search for life, we use potent instruments, for example, telescopes, that approach the search from distance, for example, by the orbit around the planetary body, searching for the presence of certain characteristics like gases or certain mineralogical patterns or geomorphology. Whereas in situ search for life, what we do is that using vehicles like rovers or instruments that land on the rocky planet or whatever the place, and they either collect physically samples or just analyze, but in situ analyze characteristic features or properties. of the place where they are. Well, in this varied scenario of possibilities, the search for life from the lipid biomarkers perspective requires the physical analysis, the physical handling of samples, and involves several disciplines that at least involve biology, chemistry, and geology. Biology, because lipids are structural components of life, as we will shortly discover. Chemistry, because they are chemical compounds whose properties and reactivity are governed by the principles of organic chemistry. And geology, because they are composed of very resistant skeletons of hydrocarbons, which tend to last a lot of time, and they can be measured in geological scale, like billions of years. So accordingly, lipid biomarkers constitute a useful tool for the search for life because they are able to trace it back to very early times in the history of life on Earth. In addition, due to their ubiquity in the environments in life on Earth, they involve not only the very biosphere, but also some of the other spheres that govern the planet, like atmosphere, lithosphere, or hydrosphere. And even though to date, there is still no clear definition for life, what seems to be a consensus is that there are a number of criteria that life must meet to consider it like that. And that's the capability of replication, of growing, and confinement. Replication, which means to have offspring that carry some of the same genetic information. Growing, which means performing a metabolism that transfer information, and the property of encapsulation or confinement to isolate all this genetic and metabolic material isolated from the surrounding environment. All these three properties are carried out by three monomers that allow life to exist through ensembling or polymerization, and they are the so-called building blocks of life, which are the nucleic acids for the case of replication, amino acids for metabolism, and fatty acids, although we will see that there are some other compounds, but basically they are fatty acids for encapsulation. And because of the different chemical characteristics and functions that these three monomers or building blocks have, they also show different specificity with respect to the original source that they come from, and also in relation to the resistance to degradation, in a way that we may find this sort of transect from an end member of highest specificity, taxonomic specificity related to the origin, in the case of nucleic acids, towards the highest recalcitrance or resistance to degradation for the case of the fatty acids. And the convenience of having one or another property, specificity, or recalcitrance will depend on the aim of our study. So in the case of the search for life as a general question, I think that recalcitrance or chemical resistance may be a quite convenient feature to pursue. In any case, the three kinds of biomolecules are intimately related by their functionality in the cell, which is the basic unit of life as we know it, because we know on the Earth, all known life forms, from the simplest to the most complex one, all of them are composed of basic units that confine inside the genetic and metabolic material, which is necessary for the growth of the cell, isolating it from the surrounding environment. And the physical basis of such cell membrane is a lipid bilayer, which is segregated by the internal part of the cell from the external towards the interior, from the external environment towards the interior of the cell, which in turn will carry it intercalated within this lipid bilayer some other of the biomolecules, let's say carbohydrates, proteins, and other metabolic components, which are involved in the exchange with the outside environment. But what exactly is a lipid bilayer? What do we call lipid? Lipids actually, they don't have a very clear and unique definition as a term lipid. They are generally considered structural components of cell membranes, but very often they are just methodologically defined as those organic compounds that dissolve in organic solvents instead of aqueous media or water. Although the main role of them are the barrier function that I've just mentioned, Lipids are also involved in many other membrane functions, such as providing a stabilizing matrix for proteins, transport of nutrients into the cell, and intercellular membrane trafficking. They are also responsible for the maintenance of the protonmotive force. They are used as energy sources, like, for example, the triacylglycerols, but they are also involved in the regulation of the membrane fluidity, which they get to control by modifying their configuration according to external conditions, such as pressure, pH, or temperature. In any case, it is the widespread presence in the cell barrier of all known life forms on Earth what makes them ubiquitous biomarkers, at least on Earth. But we hope that it will be the case also beyond our planet, because whatever the type of life we have, this life must have a somehow container that isolates the life material, however it is, from the surrounding environment. So we hope it will be something very similar. So hopefully we can consider it an ubiquitous biomarkers overall. On Earth, the majority of membrane lipids are glycerol-based. They contain this molecule that I mark here in blue, and then they have hydrophilic polar head groups, which are hydrophilic. That means that they love water, and they are orientated towards the intracellular and extracellular aqueous media. And then they also have two hydrophobic tails, which, in contrast to the hydrophilic head group, they fear or they hate, let's say, water, and they build up the physical basis for the lipid bilayer. These head groups of lipids are typically phosphate-based. They have a phosphate compound, but they are also common, containing instead glycosidic, amino, or sulfate-based head groups. And then the two hydrophobic chains are connected to the glycerol backbone through two types of bonds, depending on the organism we are talking about. If we talk about archaea, these bonds are ether-based, which connect the glycerol group with the rest of the chain, which has this isoprenoidal configuration, this type of hydrocarbons with a lot of branches, methyl groups, handing from the backbone. And they tend to form mono or bilayers, depending on if they have 4 ether groups or just 2. In the case of bacteria and eukarya, both of them have instead ester bonds, that connect the glycerol groups with the rest of the chains, which in this case are, instead of this isoprenoids chain, they have fatty acids of this configuration, and these are the ester groups. And in turn, they differ in the type of chains that they have, I mean bacteria and eukarya, both in length and in the content of insaturations, which are double bonds. Typically, eukaryotic lipids, fatty acids, they are longer in chain, and they tend to contain more than one double bond. They are polyunsaturated, we said. And usually they have even numbers of carbon. Whereas in bacteria, the chains are shorter. They tend to be between typically 14 to 24 carbons. Instead, they contain only one double bond. They are monounsaturated. And they very often also contain a methyl group, a branch that we call in penultimate or antepenultimate position, what we call iso or anti-iso configuration. Then some examples that you can see for the free lipid biomarkers that we obtain when we analyze a sample upon hydrolysis of these ether or ester bonds are some of these that I show you in the screen. For bacteria and eukaryotic cell membranes, typically we have saturated alkanoic acids, like this one in the screen, that it's monounsaturated, typical from bacteria. Then in both cases, bacteria and eukarya, we can find what we call normal alkanoic or alkanoic acids or alkanols, which are linear unsaturated chains with an hydroxyl or carboxyl groups, which over time and upon the functionalization, degradation, they tend to lose these functional groups and become just the skeleton of the hydrocarbon, which we call normal alkane. In the case of archaea, instead, we typically find these isoprenoidal structures with a lot of ramifications. The typical biomarker is archaeol, which gives name for the organism, which upon degradation can also lose these ether bonds and become a more simple chain of hydrocarbons with branches, like this phytane that I show in the slide. Other type of lipid biomarkers are cyclic isoprenoids that I previously mentioned that may confer, or they can modify the fluidity of the membrane because they confer rigidity to the cell membrane by introducing this type of semiplanar compounds, cyclic compounds that typically are hopanoids in the case of prokaryotes or esteroids in the case of eukaryotes. In both cases, they are very common, introduced into the cell membrane and change the disorder configuration that the cell membrane may have in certain conditions. For example, at high temperatures, that the fluidity of the cell membrane is excessive. By introducing these cyclic polycyclic compounds, we can get a more ordered view like that we can see at the lower and right part of the slide. And then another type of compounds considered lipids are the pigments. Pigments are those compounds involved in the interaction with light, either to transform it to chemical energy, such as the case of chlorophylls, or to protect organisms from radiation, from light, which are the carotenoids, for example, and also other compounds that tend to induce reactions or behaviors. Well, depending on the technique the analytical technique that we use to analyze these compounds, we could be able to detect the whole complex molecule, which is, these are quite heavy and difficult to detect, but we can detect them, for example, with techniques like HPLC, high performance liquid chromatography. But if we use a more common technique for the analysis of lipids, which is the gas chromatography coupled to mass spectrometry, GC-MS, we are not able to detect such a heavy and big molecules, but rather we detect some residues. For example, in the case of chlorophyll, what we detect typically is the side chain of this chlorophyll molecule, which is called phytol, which contains this hydroxy group at the end. Upon degradation, and depending on the conditions in terms of abundance of oxygen in the environment, this phytol will become pristane or phytane. So it can inform us about the conditions where this compound was deposited or accumulated. So we can say that lipids, as other biomolecules, can be used to track biological origin, attributing their presence to certain biosources through the synthetic pathways that has produced these compounds. Even though I previously mentioned that these are not the most taxonomically specific biomarkers or biomolecules, they indeed help us to assign at least some basic origins, discriminating between general groups of organisms. For example, we can discriminate between terrestrial or aquatic biomass. Also, we can discriminate if we have a prokaryotic versus eukaryotic compound. And even more specifically, we can discriminate between general groups like animals, plants, fungi, algae, bacteria, and so on. And within some of those, we can discriminate if we have vascular plants or macrophytes, for example, or oxygenic versus anoxygenic bacteria. And when we combine the mere molecular analysis of lipids with their isotopic stable carbon isotopic composition, we can even get more information about the synthetic pathway which has provided the synthesis of these compounds. And thus, we would be able to discriminate between the major autotrophic pathways, for example, the Calvin cycle, the reverse tricarboxylic acid pathway, the 3-hydroxypropionate bicycle, or the reductive acetyl-CoA pathway. Each of these metabolisms conducted dominantly one or another type of group of organisms. So we could be able to narrow down a little bit more the biosource, the organism that has produced that compound. And to better visualize this idea, I have shown here a plot with the delta 13 value. This is the stable carbon isotopic composition of different compounds from the apolar fraction of one sample, where you can see different N-alkanes, normal alkanes, with a varied number of carbons, and some other compounds like squalene or diploptene and so on. Well, by representing here the delta-13 ratio, we can assign these values and the relative enrichment or depletion on the heavier isotope, the carbon-13, in a sample or in a compound. We can attribute it to one or another metabolic pathway, as you may see here at the right. So you can connect it with a biosynthetic pathway and, again, be able to constrain better the biosource from which this lipid has been produced. But apart from that, in contrast to other biomolecules, the additional very useful information we can get from lipid biomarkers is regarding the depositional environment in which these compounds have been either produced or accumulated. For example, from the proportion of pristane or phytane that we have in a sample, we can infer whether the biomass has been deposited in conditions dominantly oxic or anoxic. From the relative abundance of short, long-chain alkanes, normal alkanes, versus the short-chain alkanes, we can deduce if the system is more aquatic or more terrestrial. Or from the proportion of estanols relative to their parent sterols, we can infer the degradation extent that the organics or that sample has overcome, and thus know more about the preservation and the environmental conditions of where that sample was collected from. So altogether, lipids are a powerful biogeochemical tool for the search for life, with diagnosis capacity to first identify biogenicity by distinguish a biotic from an abiotic pattern. We are able to identify biosources and connect it with major metabolic features. We can trace back biosignatures in a geological scale, and thus afford paleoenvironmental reconstruction further back than with other biomolecules like proteins, DNA, and so on. We can get closer to the reconstruction and understanding of early life, and eventually we can also assess the preservation or degradation extent of the life traces. So at this point, we already have a rough idea of how lipid biomarkers may be useful in the field of astrobiology, where we said that one of the key questions is to decide whether life ever arose beyond the Earth. Specifically on Mars, we have an instrument already on Mars, the SAM instrument, the sample analysis on Mars, which is on Gale crater since 2012, analyzing samples to try to detect organics as part of the Mars Science Laboratory mission on board the Curiosity. Not yet on Mars, but on the way to go, hopefully by 2028, the ExoMars mission, which has to be postponed because of the Ukrainian war, plans to make some drilling on the Martian subsurface to try to detect traces of present or most likely past life on the subsurface of Mars, by using a more sophisticated mass spectrometer version of the one in the SAM instrument. But in both cases, these two missions involve instruments that try to measure organics similar to the lipids that we analyze, and they are very promising in detecting some sort of molecular traces of life. And apart from these two, having instruments in situ, we also have the Mars Sample Return, which is conceived to bring back to Earth a selected set of samples that have been already collected on Mars and are on the way to collect even more, to try to apply a whole spectrum of sophisticated techniques that we have on Earth to try to characterize the sample as better as possible in the search for any trace of Martian life that they may have. But the thing is that so far, apart from dichlorobenzene and a couple of dichloroalkanes, on Mars, only some sort of hydrocarbon chains have been detected, this type of N-alkanes from 10 to 12 units of carbon. And the thing is that although they have been hypothesized to potentially originate from carboxylic acids and hopefully have some connection with a potential life. The thing is that these kind of structures are very unspecific, and they can perfectly come either from biotic sources or from abiotic processes. So in order to point at a more likely biosignature, we need to keep searching further in order to try to detect more specific biogenic features, like hydrocarbons containing, let's say, some double bonds or insaturations, some branched chains in specific positions characteristic of lives, like the iso, anti-iso configuration, at least some functional groups like hydroxy or carboxylic. And even better would be to find one of these guys that I show in the left corner of the slide, like an opanoid, which could be more likely connected with a biotic origin. In addition, in meteorites, longer chains of N-alkanes have been detected together with some short carboxylic acids and even pristane and phytene. But in all cases, in all specimens, all these hydrocarbons have been connected with some sort of terrestrial contamination through the trip of the meteorite towards the terrestrial surface, either with that or with a biotic generation of the hydrocarbons through, for example, Fischer-Tropsch reactions. In this case, the discrimination between biotic or abiotic sources for these hydrocarbons has to go largely through scrutinizing the molecular distribution of these hydrocarbons, because the synthesis of these hydrocarbons differs very much when the process is abiotic, where the carbons are added one by one, whereas in the case of biogenic hydrocarbons, in typical enzyme-driven biosynthesis on Earth, they are typically added by pairs. So they both show very different molecular patterns when we see the whole distribution. And this can be combined with isotopic analysis of the compounds. So if we detect the carbon-13 depletion in alternating carbons, that could be a more solid clue for the assignment of the hydrocarbons to biotic origin. But another less explored option would be to study the chirality of hydrocarbons, which, although it's not very much known, it is a very useful approach or technique, because some hydrocarbons, like branched hydrocarbons, such as those that you may see in the left part of the screen, they have, despite lacking functional groups, they contain quaternary carbons that have very different branches. The 4 branches are different, so they provide multiple stereocenters. And by studying their chirality, we can infer a biogenic origin, which may be very useful in the search for life. The good thing is that this type of chiral hydrocarbons are present in biology, for example, in the form of archaeol, which is typical from archaea, we said, but also some other compounds coming from phototrophs like phytol and their derivatives, pristane and phytane, and also tocopherols. The not so good thing is that these compounds have very small specific rotation, and their configuration assignment is quite an analytical challenge. And this is probably the reason why there are just a few groups investigating on this field. One of them is the one led by Uwe Meyer-Heinrich in the Université Côte d'Azur in Nice, in France, who have got to separate by regular chromatography just by using a chiral column. They have got to separate 2 enantiomers of some commercial compounds, but they need to work harder to get that separation in longer chain of compounds. One of the strengths of this option is that these branched chiral hydrocarbons tend to racemize and degrade slowly, so they are more resistant to degradation than the regular N-alkanes, and thus offer a promising alternative to identify hydrocarbons' biogenicity with application, for example, for astrobiology. And the rationale behind it is that, similar to amino acids, the hydrocarbons coming from life, they tend to have an enantiomeric excess. And given that they have a greater stability compared with other biomolecules like amino acids, they would be able to be detected, to resist and be detected for longer periods of time. So if life ever arose on Mars, and if it was similar to the life that we know from Earth, we can assume that the derived hydrocarbons could bear a similar enantiomeric excess that could be detected for longer periods of time, since life could have emerged on Mars at the early scenarios on that planet. And another good thing of these chiral hydrocarbons is that as hydrocarbons, they don't need to be derivatized, as it happens with amino acids for their analysis. So this facilitates their analysis in situ. And this brings us to the point of how do we analyze lipids. I already mentioned some of the techniques used for the molecular analysis of these compounds. But before that, we need to extract the organics, potentially from biotic origin, lipids. So for that, what we do is to subject the sample to an extraction process with organic solvents, let's say methane, acetone, dichloromethane, many others, depending on the protocol we use. Then we obtain a total lipid extract that we need to concentrate for analysis and purify. And also, depending on the protocol we use, we may want to separate this total lipid extract in different fractions to facilitate our analysis, and these fractions depending on the polarity of the compounds. So after that, we analyze the different fractions in two ways. Well, we can do it in one or another, but we used to do it in two ways. First, the molecular characterization based on the use of a gas chromatography mass spectrometer, which accomplish the identification of the chemical structure. So which compound do we have? And then in parallel, we can couple this GCMS to an isotopic ratio mass spectrometer, so we can measure the stable carbon isotopic composition in order to get another clue, an additional clue, to know more about the origin of this compound. And now that we have a rough idea of what are lipid biomarkers and what they can be used for, I would like to give you a brief glimpse of the type of research lines that we conduct at CAP in terms of lipid forensics. We, in synergy with other research groups at CAP, we explore extreme environments to characterize extremophilic life from a lipidic point of view, searching for molecular and isotopic fingerprints of life in harsh and challenging environments that very often keep some analogy to some of the major astrobiological targets that we have in astrobiology, like Mars or the icy moons. We also reconstruct biosources and environmental conditions in ancient scenarios based on the chemical resistance and great preservation potential that the lipid biomarkers have. We investigate how lipids get altered over time, trying to know when and how do they degrade and where and upon which conditions do they get preserved, what it's called the taphonomy of the lipid biomarkers. And in a way or another, we accomplish both fieldwork studies and lab-based experiments to understand preservation patterns of these biomarkers, lipid biomarkers, trying to detect their fingerprints of life when life is scarce, with the ultimate aim of learning what, how, and where to search for life beyond our planet, beyond the Earth. Here you may see some of the extreme environments we have been exploring, from cold and dry environments in Greenland, Antarctica, or Iceland, to hot and hypersaline deserts like the Atacama Desert or the Makgadikgadi salt pans in Botswana, passing by high-altitude hypersaline lakes in Argentina or in the Indian Himalayas, hydrothermal regions in Chile or Iceland, hyperacid rivers such as Rio Tinto here in the south of Spain, or even polyextreme environments in the Dalol region in Ethiopia. And just a few examples of the studies we conduct, for instance, to identify biogeochemicity in astrobiologically relevant environments. such as serpentinization-associated systems, like this study, where we study one region in Spain that contains serpentinization systems in the subsurface. We studied 7 groundwater systems from the Ronda region, which is one of the largest outcrops of subcontinental peridotites. We compared the organics that we collected from the groundwater in these 7 sites, and compare that with the organics that the associated precipitates of carbonates they had in the surface of these springs. And we were able, because of the molecular distribution patterns of both type of samples, groundwaters and the associated travertines, we were able to infer different origins, dominantly abiotic in the case of the groundwater versus dominantly biotic in the case of the associated carbonates, precipitates. And we were able to propose these travertine precipitates as spatiotemporal archives of that tend to amplify the scarce biogenic signals that we have in the groundwaters, which may be very useful to identify serpentinization-associated systems in analogous environments on Mars, for example. In this other study, what we investigated was the lipid biomarkers in 2 submarine coal sieve systems, where there was intense rock-fluid interaction, and maybe interesting to study the carbon sinks and carbon cycle in systems which are analogous to the icy world's oceans. We studied 2 types of carbonates, autogenic carbonates, in 2 systems, one clatrite in the Pacific hydrate ridge, and another carbonate from a submarine chimney in the Gulf of Cadiz in the Atlantic Ocean. And by the molecular and compound-specific isotopic composition of the lipid biomarkers, we were able to distinguish 2 different origins of the carbonates in both systems. In the Pacific clatrite, we observed that the carbonate stemmed from biogenic sources, biogenic methane, which was oxidated anaerobically by a syntrophic action of methanotrophic archaea and sulfate-reducing bacteria. Whereas in the carbonates from the Atlantic, from the chimney, we could see that the carbonate came from a combination of abiotic methane, thermogenic methane, and allogenous carbon coming from pelagic biomass, which was settling down from the water column. We discussed these results in the light of potential similar carbon cycling pathways in analogous icy moon oceans, and we showed how the isotopic analysis of carbon may be crucial for detecting biosignatures in icy world carbon sinks. And another last example is this one that we conducted on the Atacama Desert, when we compared the lipid profile of 3 different rocks from the Triassic-Jurassic times. And we were able to reconstruct 3 different scenarios of deposition in the 3 systems of about 200 million years. And we were able to distinguish, apart from the biosources and prevailing metabolic pathways, we were able to reconstruct different redox conditions in the 3 systems, different ecosystems which had been developed in terrestrial versus aquatic conditions. And by comparing our results based on lipid biomarkers with those obtained by using another biomolecules like proteins and immunosignals, we were able to demonstrate how extraterrestrial life detection can benefit from the use of different biomarkers to overcome diagnosis limitations due to the lack of specificity or alteration over time of one or another type of biomolecule. And just to finish, I would like to end mentioning just some of the work we have started to develop recently, which tries to monitor what happens to organic compounds, potentially of biological origin, when they are subjected to radiation at different control conditions of different doses and the different conditions, in presence and in absence of certain species that may be conductive to further degradation, like oxidants, like perchlorates. And the idea behind it is to understand the taphonomic processes that putative biomarkers may undergo in the space, and try to define the patterns of the functionalization and loss of complexity during these radiation experiments, ultimately to learn to recognize molecular residues of possible extraterrestrial life in future missions outside the Earth. And with this, I will stop here, not without remembering and mentioning the rest of the lipid guys that work with me in this lipid biomarker, in this group of lipid biomarkers in planetary exploration, where you can see which are the fatty guys that are working presently at CAP. Pablo de Lucía Finkel, which is a PhD student with me in the Department of Molecular Evolution, and Daniel Carrizo, who is a researcher at the Department of Habitability and Planetology. And just recognizing some of the groups and institutions with whom we have collaborated in a way or another, just sharing either samples, field campaigns, ideas, or projects. So that's all. Thank you very much for listening.